Previously on Secret Singapore, follow us as we find ways to enter some rarely seen limited access labs where personalized and predictive medicine is designed and created. Germs sometimes reach a point when they beat the drugs that are designed to kill them, a situation known as antimicrobial resistance or AMR, and the lab is dedicated to addressing this. Called the Smart AMR IRG, it is a translational research and entrepreneurial program that applies theoretical discoveries to further develop practical applications. It is headed by Dr. Megan McBee, Senior Scientific Director.
Now, there's a growing problem of antimicrobial resistance, so that means that the bacteria are no longer killed by the drugs that we're giving them. And therefore, we need to develop new drugs that will kill the bacteria better or help our body kill the bacteria better.
Why does it happen that the drug that used to kill bacteria no longer works?
It can be many things, such as the bacteria develop ways to pump the drug out, or their genetics change so that they no longer need whatever it is that the drug is blocking in their system.
So they have evolved?
Yes.
Is that
Yeah, I mean, the resistance, it's usually they acquire it from another bacteria that may have already evolved and already has this resistance, or they evolve themselves. So they change, and that allows them to survive in the presence of the drug.
Okay, can you walk me through the different things that you do in the lab?
Right, so we do a lot of different activities looking at evaluating the bacteria, right? So we can evaluate the different chemical compositions that are in them. So we use very fancy machines, mass spec analyzers. We can do a lot of high throughput, either doing sample prep or screening for drugs, whether the bacteria survive or are killed by different drugs, to look at whether we have something that might be able to be taken to a clinic and hopefully maybe one day be a drug to treat people.
And of these researches that you're doing, which one is close to being produced or used in a drug?
So a lot of our work is still early stage in terms of drug development, but we have some that are further along in the discovery process. So several of those, we have one drug that's been identified that helps your body actually respond better to a bacterial infection. So you can give that drug with a existing antibacterial drug, and then together they kill the bacteria much faster in the presence of your immune cells and your body. There's also a couple other drugs that were existing for, say, bacteria Y, and we think that now they can actually be used in combination with an existing drug for bacteria Z.
Okay.
So that's a project that's pretty exciting. Yeah.
Smart AMR works collaboratively as one research group and leverages the scientific and clinical strengths of MIT and Singapore. It is among the few labs dedicated to both AMR-related research and innovation. Smart Camp IRG is helping provide more patients with access to next-generation cell therapies, the use of living cells such as immune cells and various stem cells as medicine. Their work requires not only improving the efficiency of manufacturing current cell therapies, but also exploring new avenues for improving cell-based approaches in medicine. Professor Jong-Yoon Han, a professor of biological engineering and electrical engineering at MIT, is the co-lead principal investigator at Smart Camp.
Cell therapy already curing some of the uncurable patients here in Singapore as well as globally. And we are a group of MIT engineers coming to Singapore and working with local universities and US and TU, as well as hospitals and research institutes like A-STAR, to think about innovative ways to make these medicine much more economical, accessible, and much more safe. We making the cell therapy more economical, and also maybe having a much more regular quality and function. And then thirdly, actually, we ensure their safety of the cell therapy. We specifically look into the patient cell harvesting process, which is often called leukapheresis. That currently involves a lot of manual operation and manual separation processes. So we maybe introduce some microfluidic innovation so that we can render that process more automated and faster and more efficient. The second area we work on, right now, we don't really have a good measurement to really reveal that, okay, this particular batch of the product is a good quality. So we work on many new technologies where we can take a look at the cell and maybe return a measurement which can reveal something about the quality. In a layman's term, what we are doing is a little bit of MRI scanning on the cell. So without disrupting the cell, we may be able to reveal what kind of a cell identity this particular cell group has. And if that identity is deviating too much from the original prescribed range, then we know that this particular batch may not be of the right quality to be injected. Finally, the safety of the cell product is very important. And for that, when the cell production process sometimes subject to some what we call as adventitious agent, such as a virus or bacteria growth, that's going to be a big concern and need to be detected. We are developing some machine learning enabled techniques and detection methodologies to really detect event of such a contamination much more faster, preferably within a day, so that we can speed up the quality and safety validation process before we actually inject these products into the patient. We tend to generate very new idea, and what that means is that we really need to validate these ideas in our lab. But even if these ideas are validated in our hand, we still need to bring this thing through multiple layers of validation and checkup until we are confident that this actually can be deployable in the manufacturing space or environment. We hope to work more with our partners in the next 1 or 2 years, and maybe we have enough confidence and validation to start applying this idea in actual realistic situations.
The group collectively tackles the challenging problems of the industry that does not belong to any one department or discipline, as Professor Han says. Every researcher at camp is challenged to go beyond their comfort zone in approaching topics and issues that may lie outside their field of expertise. This intersectional, interdisciplinary collaboration and research is what makes camp truly unique. Next up on Secret Singapore, we visit a specialized lab that analyzes and keeps stool samples for nutraceutical and medical use.